The intersection of mitochondria, lysosomal disease, and the impact on multiple body systems, including the nervous system, is absolutely fascinating, and I hope you'll think so after you hear these talks today. I'm very happy to welcome our moderator for today's session, Professor Alexey Pshezhetsky, who directs the Medical Genetics Diagnostic Laboratory and the Alyssa Linton Laboratory of Lysosomal Biology at St. Justine Research Center in Canada. Among his many research interests, Dr. Pshezhetsky has pioneered characterization of the mouse model and research into Sanfilippo type C, including examination of mitochondrial dysfunction. Now I'll hand it over to Dr. Pshezhetsky to start us off with an overview and followed by an introduction of the other speakers. Thanks.
Hey, good afternoon, everybody, and thank you so much, Kara, for this kind introduction, and also thank you for inviting me to moderate this session at this very successful and exciting conference. And I think the subject of this session on mitochondrial involvement in the Sanfilippo pathology is very, very important. And we will hear from two speakers today, from Dr. Rebecca Ganetsky, who is assistant professor of pediatrics in Children's Hospital of Philadelphia. And her research focuses on the mitochondrial health and bioenergetics in the context of rare genetic metabolic disorders. And then our second speaker would be Dr. Philippe Gordts, who is an associate professor of medicine in the Division of Endocrinology and Metabolism and Glycobiology Research Training Center at the University of California in San Diego. And Dr. Gordts, who was trained in Belgium and then came to California, he leads a group of scientists studying the heparan sulfate, proteoglycans, and the milk oligosaccharides in the context of cardiovascular and metabolic disorders, including the diabetes, atherosclerosis, and obesity. So, but I kind of asked to do a short introduction to this session because I think we now underestimate the role of the mitochondrial dysfunction in the lysosomal disorders. And if we think that lysosomes are recycling plant of the cell, while mitochondria is an energy plant, and of course, we can do much more damage by destroying the energy plant. And when you look at the PubMed, you see only about 10 papers on this subject, but it starts in the 60s, where the abnormal, morphologically abnormal mitochondria, referred to as mitochondrial buds without morphological similarities with mitochondria, were identified in the biopsy materials. And it was proposed that they are raised by direct transformation of a mitochondria. Next slide. Next slide, please. Yes, thank you. So then in the 90s, still the last century, when people started looking more at the specific materials accumulating in the neurons and in the brain of Sanfilippo patients, they identified abundant mitochondrial enzyme, which is mitochondrial ATPase synthase, specifically its subunit C, as the materials that accumulates in the brain. So they reported increased immune reactivity for this ATPase synthase subunit C. And if we look next slide, in the brain, specifically in the mouse models, you can see this is a mouse model of the Sanfilippo type C. You see this layer of the neurons, deep pyramidal neurons of the cortex, somatosensory cortex, that are very abundant for this subunit C of mitochondrial ATPase synthase, or SCMAS. And next slide. You can see also the same accumulations in the other Sanfilippo mouse models. This is work of Elizabeth Neufeld, and you see in 3A, in 3B as well. And they also showed that this is the biomarker that responds to the gene therapy. Next slide. Then all this, why this abnormal mitochondria accumulate and why the subunit C misfolded accumulates became clear after this seminal paper that came in 2008 from the work from the lab of Andrea Belabia, where they identified the deficiency in the outer phagosomal function of the cell, specifically caused by the inability of autophagosomes to merge with the lysosomes. And then they concluded that this results in the massive accumulation of dysfunctional mitochondria. And they proposed that this dysfunctional mitochondria, this process, can be putative mediator of the cell death. So next slide. This becomes really clear how the lysosomal dysfunction and how the dysfunction of autophagy results really in the disrupted mitophagy, increased mitophagy flux, and mitochondrial perturbation. So next slide, we looked at the 2 mouse models of the Sanfilippo type C, and next slide, we could find this abnormal mitochondria that completely miss the internal structure, and they are clearly dysfunctional in all the types of neurons. And also on the next slide, when we looked at the mechanism underlying the synaptic defects in Sanfilippo syndrome, next slide, we found when we analyzed the synaptosome by proteomics, and we found that the most reduced class of proteins, of course, are synaptic proteins, but the second class that is strikingly reduced in the Sanfilippo mice are mitochondrial protein, and that includes, for example, malate dehydrogenase, mitochondrial, the dynamine-like protein that is important in the separation of mitochondria, succinate coenzyme ligase, and so on. So next slide. You see this abnormal mitochondria not only in the neurons, but also more recently we found them also in the oligodendrocytes that make myelin layer of the neuronal axons. So on the next slide, when you look with time at the progression of these enzymatic deficiencies in Sanfilippo mouse brain, you see that the enzyme activities such as citrate synthase, coenzyme Q reductase, other most important mitochondrial enzymes, they go down with time, whereas for the wild type mice, they're not decreasing. And that's at the end, you have pronounced mitochondrial deficiency. So last slide, it all allowed to hypothesize that mitochondrial damage is actually a very important part of this multifaceted mechanism underlying brain disease in neuropathic MPS. And today's session will see how this progressed most recently. Well, thank you very much. So now to Dr. Ganetsky.
Hi, thanks very much for having me. I am going to talk to you a little bit about mitochondrial dysfunction in Sanfilippo, but unlike Dr. Pshezhetsky, I'm not an expert in Sanfilippo at all. And in fact, what I know about mitochondrial dysfunction in Sanfilippo syndrome, I largely am citing from his papers. What I really do want to focus on is mitochondrial dysfunction, what that means, how we measure it, and what we can do about it, which is really my area of expertise. And I'm a physician scientist at the Children's Hospital of Philadelphia, where I develop biochemical tests to measure mitochondrial function for patients. Next slide, please. So we're going to talk about what mitochondria do for us, the clinical tools that we have available, as well as tools that are emerging into the clinical space that analyze different parts of mitochondrial function, how Sanfilippo can interact with mitochondria, and then what tools we have to treat mitochondrial dysfunction. Next slide, please. So we'll start by talking about what mitochondria do in our bodies and what their normal function should be to help us understand how dysfunction can cause disease. Next slide, please. So mitochondria are subcellular organelles. And here I have 3 different ways of looking at the picture of mitochondria. On our bottom right is what you usually see, that cartoon bean of the mitochondria. And what that shows you is that mitochondria have 2 membranes, an outer membrane and a very kind of convoluted inner membrane. We call that convolution cristae formation. And within those cristae, in the convoluted area, that pink area in the middle is called the mitochondrial matrix. The mitochondrial matrix is home to most of our metabolic enzymes. So mitochondria are organelles inside our cell. There's 100 to 1,000 of them in each cell. And so what you see above the pink bean on the top right of your screen is a cell that's been stained with what's called Mitotracker red. Mitotracker red stains mitochondria red. I know. That circle in the center is the nucleus. The nucleus of the cell doesn't have any mitochondria, and then you see around it all those red dots, which are the mitochondria forming networks within them. So mitochondria take up a lot of space in the cell. They're dynamic. They need to move around the cell. They take on different sizes and shapes, and those different sizes and shapes are actually really essential for their function, which is what I'm showing you in the bottom left-hand corner. The reason that I emphasize so much how many of them there are, the way that they're shaped, and how they move around the cell is to emphasize to you that when there are other things that are taking up space in the cell or distorting the ability of those mitochondria to form networks, that can be one mechanism of disease. Next slide, please. So what do mitochondria do when they're functioning? There's a lot of mitochondrial metabolic pathways, but this is kind of what we call central metabolism. And central metabolism, which takes place in the mitochondrial matrix, and what I've shown here on the left is the cytosol, the rest of the cell. And the right is the mitochondria. And in the mitochondria, up is the intermembrane space between those 2 inner and outer membranes, and down is the matrix, the heart of the mitochondria. In the cytosol, you can turn glucose into pyruvate, and that produces a little bit of energy in the form of ATP and a little bit of reducing equivalents in the form of NADH. But inside the mitochondria is the TCA cycle, where most of our nutrients are oxidized, the fatty acid oxidation cycle, where fats are oxidized, and those things produce a lot of reducing equivalents in the form of NADH. NADH feeds complex 1 of the mitochondria. The oxidation of NADH and then the subsequent oxidation of coenzyme Q is coupled with the pumping of protons against their gradient across the mitochondrial membrane into that intermembrane space. They don't like to be against their gradient, and so that results in a potential energy, that desire of the proteins of chemistry to return to equilibrium. And so that complex 5 of the mitochondria, the yellow complex, ultimately allows those protons to flow back and along their gradient. And that release of potential energy is coupled to the production of ATP, so energy in the form that the cells can then use. Not over 90% of cellular energy is made in the mitochondria, and that's its central role, but that's not its only role. Next slide, please. When we talk about mitochondrial dysfunction, this really is the focus. There are other ways that mitochondrial can be dysfunctional, and we'll talk about some of them. But the kind of core things that we clinically look at when we're looking for signs of mitochondrial dysfunction all come from here. They come from things like there is not enough oxidation of NADH to restore NAD+. So the reduction oxidation balance of the cell is imbalanced in favor of the accumulation of NADH. That means that other reactions that use NAD+ to make NADH, which are the oxidation reactions, burning fuel for energy, those become inhibited because of the accumulation of NADH. The pumping of protons against their gradient is coupled to the capture of electrons. This is also called the electron transport chain, as you might remember. And those electrons are supposed to flow from complex 1 to complex 3 to complex 4. And if they don't flow along their gradient, that leak of electrons, especially from these arrows are supposed to show you that, especially from complex 1 and complex 3, cause the generation of reactive oxygen species, really free-ranging electrons in space that cause damage to the cell. So that's another thing that we can look at. And finally, the goal is to make energy in the form of ATP. And we can look not directly at how much ATP is produced. We wish we could, we can't, but at biomarkers of energy production in the cell. Next slide, please. So what are things that we can test for biochemically? We can measure metabolites in blood and urine that help us increase the index of suspicion of mitochondrial dysfunction, but may or may not look directly at those things that I showed you. We can actually directly measure the function of the electron transport chain enzymes in muscle or other tissues. That's very specific. That really asks the question, how well are the mitochondrial enzymes working? But it's invasive. And when we're talking about something where we suspect secondary mitochondrial dysfunction, like Sanfilippo, it may be too specific. It may be looking too closely at how each individual enzyme is working and not asking the big picture question of, are the mitochondria as a network working well and healthily in the cell? The other advantage of these biochemical tests that we have available is that when we talk about therapies, supplementing nutrients that have become deficient because the mitochondria aren't working, like coenzyme Q10, or trying to fix the redox balance of the cell, or trying to reduce oxidative stress, these biomarkers can be used to monitor that response to therapy. So what are these biomarkers? Let's take a look at them on the next slide. So the historic gold standard, and I say this just because every time I give this talk, people say, well, what about muscle biopsy? The historic gold standard was muscle biopsy. And what I'm showing you here are ways that we can look at mitochondria in the microscope to see that they're diseased. But this is very, very specific, not just to primary genetic mitochondrial diseases, but to specific mitochondrial diseases. Not all mitochondrial diseases have these abnormalities on microscopy. The other thing that we can do with a tissue biopsy is we can look at the individual enzymes, and we can also look at how they work together to consume oxygen. So next slide, please. So looking at them in an integrated fashion is called oxidative phosphorylation analysis, or polarography, and it measures an integrated function of how they work together. It's a little bit less traditional than enzyme testing for the enzymes in the electron transport system, but for something like Sanfilippo, where we think that there may be a secondary dysfunction, it's better because it looks at how the functions of the mitochondria work together to make energy. It is based on the assumption that the consumption of oxygen by the oxidative phosphorylation system is a marker for how well those enzymes are working to make energy. We do this as a clinical test here at the Children's Hospital of Philadelphia. We're actually the only center in the United States to do it, although there are European centers that do it. We do it on fibroblasts, which is skin tissue. And what I'm showing you here is that we feed each complex, complex 1, and then complex 2, and then we inhibit complex 1 and then complex 2 to see how individual complexes work. And so what I'm showing you here, this is a run from my lab. In red is a normal healthy cell line. And what you're looking at is the rate of oxygen consumption over time. So first we look at how the cells just are consuming oxygen. And then we feed complex 1 and they're like, oh, we're feeding complex 1. We should be making energy. We're going to consume a little bit more oxygen. Then we feed complex 2. These are the arrows in order if you want to follow along. It goes up. And then actually we take off complex 5, which is the slowest. And we just let protons flow freely along their gradient. So then we're letting things go as fast as they possibly can. And it goes even faster. And then we inhibit complex 1 and inhibit complex 2, and it goes slower. And so that way we see both how it's working together, and we can also kind of see how each individual complex is contributing. And black is a patient with primary mitochondrial disease. And what you can see is that they're consuming much less oxygen at baseline and then at each step along the way. And so we can really identify, baseline is 50% of control and complex 1 is 50% of control, but actually complex 4 is like only 30% of control. So complex 4 is more effective at mitochondrial dysfunction. Next slide, please. The alternative is looking at electron transport chain enzyme activity. So that measures individual complex functions. So what I'm showing you here on top is complex 1. What we would do is we'd feed something like malate or NADH, which are the substrates of complex 1. And we would measure the reduction of coenzyme Q, which is what I'm showing you in this arrow with the circle in red. I also am showing you just to remind you that that's not the only way that complex 1 makes energy. The real way that complex 1 makes energy is pumping protons against their gradient to ultimately feed complex 5. But that's not something we can measure. So we're instead measuring the reduction of CoQ. So this is really looking at zooming in on one enzyme. And the problem with this is that what I'm showing you on the bottom is there's really a continuous spectrum from normal to abnormal. There's not one correct cutoff. There's a lot of variability in the normal healthy population. And so it can be really hard to know what to make of one person in isolation. Next slide, please. Getting muscle is a big deal. Kids and anesthesia, especially kids with metabolic diseases and anesthesia, can be really challenging. We have lots of other parts of our bodies that make energy, like blood and skin. And so one really important research question is, can blood and skin be better substitutes for muscle biopsy? As I said, our test that does polarography uses skin cells. Skin has not as much mitochondrial activity, so it may be a little bit less sensitive. We think it's worth it because it's easier to get. Blood has more mitochondrial activity, but I can't get, 40 milliliters of blood off of someone, and it doesn't keep growing in culture the way that skin does. So it's more challenging. So what this is showing you here is a paper that shows that there is strong correlation from muscle to liver to blood to skin. This is really important specifically for Sanfilippo because a lot of the studies on a research basis that have been done have actually been done on brain cells and culture. And brain is obviously not a tissue that we want to look at clinically, especially serially to see if it's improving with treatments. And so knowing that there is some amount of correlation is really helpful. Next slide, please. What about biomarker screening? So these are some routinely available biomarkers that are often used in the mitochondrial community. Things like looking at elevated alanine as a marker of what the lactate has been over time, looking at elevations in glycine because glycine is broken down in the mitochondria, so that's a good biomarker for mitochondrial dysfunction, looking at how the TCA cycle is progressing and whether things are failing to be oxidized, looking at ethylmalonic acid as a marker of the disulfide redox relationship in the mitochondria. Dicarboxylic acids are a marker of fatty acid oxidation. And then looking at the acylcarnitine profile as well to see that there's delays in oxidizing the nutrients that are that form the acylcarnitines. Next slide, please. Although those tests are widely available, actually physicians who do primary mitochondrial medicine still don't agree if any of those are helpful. And what you see here is a study that I love from my partner, Sumi Parikh at the Cleveland Clinic, who interviewed 32 people who consider themselves mitochondrial experts. And he said, what test do you actually find helpful? And what you see is everyone agrees that lactate is a good marker. Lactate, I'll show you on the next slide, goes up when your NADH is not being used. And so that's what it's a marker of. But it is not necessarily sensitive and it doesn't answer a big picture question. Everyone also uses organic acids, which can show you that the TCA cycle and fatty acid oxidation is secondarily impaired. And then there's little consensus about what to do next. So there are emerging biomarkers, and actually if you advance 2 slides for me, please, thank you. And what are they and what questions can they answer? So my personal favorite, everyone's allowed to have a favorite, of the emerging biomarkers is glutathione. And that is a test that we offer here at CHOP, and it's also offered at Stanford. Glutathione is the main antioxidant in our system. And what we found in children, what Greg Enns at Stanford found in children with mitochondrial disease, over here on the right, is that kids with mitochondrial disease had less redox potential of glutathione. So what you're seeing on the axis there, in red is kids with mitochondrial disease, and blue are controls. And then what we're measuring is the redox potential of glutathione, which is a combination of how much glutathione do you have and how much of it is already reduced versus how much of it is oxidized. What he found is that kids with mitochondrial disease have less glutathione and a higher percentage of it is already oxidized because there's so many reactive oxygen species. So both the total glutathione and the oxidized glutathione are markers of oxidative stress. But not only, this is why it's my favorite, not only is that true for kids with mitochondrial disease at baseline, but if you look at the lower graph, when they are in metabolic crisis, they shift even further to the right, away from normal. And so that tells you that this is not just a diagnostic biomarker, but this tells you how people are doing over time. And I think this is a really good biomarker for secondary mitochondrial dysfunction that you can see oxidative stress. Next slide, please. Glutathione, just to say, there are other labs other than CHOP and Stanford that offer it, but it's a very unstable chemical. And so what you're seeing here in the graphs on the left is that it does oxidize over time. And so in the orange dots, samples that we had kind of left out at room temperature, you're seeing that there's already a drift in the levels. And so we found that the samples did have to be drawn into special tubes and immediately derivatized and deproteinized and frozen within about 2 hours of blood draw. And that stabilized the sample, and then it could be analyzed at any further point. Some of the commercial labs don't do that, and then the sensitivity and specificity of the test as a tracker of response to treatment is not as good. Next slide, please. There was an unbiased search for biomarkers, and that identified 2 peptides, FGF21 and GDF15. These are growth-related factors. They're neither sensitive nor specific. So this is work by one of my colleagues, Eva Morova, who's now at Mount Sinai. And what she's showing you is that in aggregate, people with mitochondrial dysfunction on the lower right have higher GDF15s, and people without mitochondrial dysfunction have lower GDF15s. But that in each individual dot, you can't really tell which group they belong to. And above is the same for FGF21. Next slide, please. And actually, our experience at CHOP found that GDF15 diagnostic performance was very similar to things like CK and lactate. It's not very specific. But what I'm showing you over on the right, and these are unpublished data, but if you look at the category of mitochondrial disease, some mitochondrial diseases, like mitochondrial DNA deletion syndrome in green, have really high GDF15 levels. And some, like complex 5 deficiency, never have high GDF15 levels, and it tends to run true in a disease. I don't know what the GDF15 levels are in Sanfilippo, but what this experience tells me is that if it translates to Sanfilippo, GDF15 will either be very useful and it'll be high in many patients with Sanfilippo, and then it'll be a marker that you can follow, or it won't be very useful, but that doesn't mean that there's not mitochondrial dysfunction. It means that someday when we understand exactly what type of mitochondrial dysfunction GDF15 correlates with, that specific subtype is not present in Sanfilippo. Next slide, please. The final thing that we can look at is the accumulation of NADH. So remember that the main way that the mitochondria make energy is oxidation of NADH in complex 1. And that returns NADH to NAD+. Lots and lots and lots of reactions in our body use NAD+. And if it's not being restored by complex 1, you accumulate those metabolites, and many of those metabolites actually we think act as signaling molecules that help our body understand that it's in energetic distress. And so they can be used as biomarkers. As I mentioned, lactate is the most common. When our body needs NAD+, it takes the pyruvate that's made from sugar in glycolysis in the cytosol, and it moves that pyruvate into lactate. That oxidizes the NADH to NAD+, restoring NAD+ for needed NAD+ purposes and making lactate go high. You can look at NADH to NAD, the direct ratio itself. And what I'm showing you here is that in fresh blood from a patient with mitochondrial disease compared to control blood in green, we do see elevations, but it's very hard. NADH is really not a clinically available test because it has to be drawn on dry ice and frozen within a couple minutes. So next slide. We can look at metabolites that use NAD+, like lactate, but not just lactate. In the mitochondria, the relationship between acetoacetate and beta-hydroxybutyrate is defined by the relationship between NAD+ and NADH. And we thought that that may be a more sensitive reaction. At the same time, Rohit Sharma found that alpha hydroxybutyrate, which is also something that can be regenerated to regenerate NAD+, was a biomarker for mitochondrial dysfunction in unbiased metabolomics. Next slide here. Oh, sorry. I apparently did not show you the data. Both of those tests are now available at CHOP, and you can order them. And we found that it's a very sensitive test for mitochondrial dysfunction. There are multiple reasons why there might be mitochondrial dysfunction in Sanfilippo, but what do we mean by mitochondrial dysfunction? What we mean may be that damaged mitochondria aren't broken down. That process is called mitophagy, and it requires the lysosome. They're not broken down well, and they continue to generate reactive oxygen species and have oxidative stress, or that you have mitochondria that are dysfunctional and are not making as much energy as they should. Next slide, please. People have also reported that the mitochondria are physically abnormal in Sanfilippo, and that they may be swollen or otherwise abnormal related to poor mitochondrial quality control. This was all done in the brain tissue. Looking at mitochondrial shape does require tissue samples. We otherwise don't have good tests that ask the question, how high quality are the mitochondria and are they diverse from each other? Next slide, please. In mice with MPS III, not the electron transport system itself, but their selection of which nutrients to use for energy are variable. And so those can, they have more lipid burning that they again think is related to less clearance of diseased mitochondria. But most of this research is very recent and emerging, and we're still learning. Next slide, please. How do we treat mitochondrial dysfunction? Can we try to decrease the primary cell consequences? Can we try to restore mitochondrial function? Can we try to help reduce reactive oxygen species, promote ATP production, and improve clearance of disease cells? So those are the things that we try to do. We try to do that with small molecules, metabolic manipulation, but ultimately diet and exercise are things that we know help mitochondria, although we don't know exactly the right ways to do that. Next slide, please. This is just a list of nutrients that we use to support mitochondrial function that are used as cofactors in the mitochondria. Some of these are antioxidants, and some of these are literal cofactors like riboflavin that help build our mitochondrial function. And these are the doses that we use in mitochondrial disease. Next slide, please. So there are many types of mitochondrial dysfunction, and we have different treatment modalities that ask different questions about how the mitochondria are functioning. Many of them are clinically available and minimally invasive. We don't totally know whether or not they'll answer questions about mitodysfunction in Sanfilippo, whether that's just in brain or whether that's elsewhere, whether the types of mitochondrial dysfunction we see in Sanfilippo can be assayed with the assays that exist. But these assays are designed to ask the questions of, are these treatments that we have working? They're minimally invasive, and so we're really in a new era of mitochondrial medicine where we can treat and then monitor the response to mitochondrial dysfunction. So that's what I had to share with you. Thank you so much for having me. And just to say, I work on a huge team, and this is everyone who works on the team with me, and thank you to them as well.
All right, thank you very much. Thank you all for attending. And unlike the previous speakers, I'm going to give you a bit more of a deep dive into how we identified mitochondrial dysfunction, at least in MPS disorders, to play a role in energy homeostasis. Because obviously, what I hope you took away from this is that they're basically the powerhouses of the cells, and anything that goes wrong with it will have significant consequences, not only the cellular level, but also systemically. So we're going to talk a little bit about adipose tissue and how adipose tissue is quite different when you have Sanfilippo. Next slide. Perfect. Thank you. So we all know that mucopolysaccharidosis are basically a conglomerate of lysosomal storage disorders, and that's important with enormous prevalence. But one of the interesting clinical features beyond the neurological features that I think is worth paying attention to and worth treating as well in the long run is the fact that many of the patients will suffer from some kind of wasting later on in disease progress and early mortality. And so the wasting is also called cachexia, and that's something you see in many different disorders, including in cancer. Next slide, please. So why is that? Why are we looking at mitochondria? Why do we think energy dysfunction is important in lysosomal storage disorders? One of the important functions of a lysosome in any cell is that it's basically a recycling center of micronutrients. A lot of the organelles that we're making, a lot of the proteins that we're making, a lot of the glycans that we're making are broken down in their individual components. And some of that will be secreted by the cell as excess. But a lot of those nutrients and macromolecules will be recycled to build up new proteins, new glycans, new lipids, and what you not. The problem with the accumulation or dysfunctional accumulation of lysosomes or lysosomal bodies is that process is now inhibited. It doesn't only affect the recycling of macromolecules, but it also affects the recycling of components that are integral to the organelles within the cells. And that is an important pathway, and we call that autophagy. It's basically the self-eating of components of the cell that you then can help to recycle and build new and healthier organelles. And we'll come back to that because that's particularly important for our nuclear power plants of the cells, the mitochondria. And when that goes wrong, that has significant consequences not only locally, but also systemically. One of the big problems when you have this recycling deficit is that you have to overcome the loss of energy that you would normally be able to rely on during that recycling process. And one of the things that I work on, and as I already alluded to earlier on, is lipid metabolism. And lipid is one of the molecules that will give you per molecule the greatest amount of energy in the form of ATP. And so we were trying in a investigative study to assess if lysosomal dysfunction would have a systemic impact on the way our bodies handle lipids as nutrients, and will they or were they going to fuel the energy deficits that we see in the cells that suffer from lysosomal storage disorders. So next slide, please. And one of the interesting reasons we were doing this is because we noticed that in a lot of the kids that suffer from Sanfilippo, and this is MPS IIIA patients, that they at later stages in life had the inability, it seemed, to gain weight. Initially, the weight gain was quite okay, but then over the teenage years, there's a significant drop-off, as you can hopefully notice here at the left of our screen, where you see the weight gain for an average boy with MPS. And if you look at the BMI, because obviously weight is affected by how tall you are, you can see that the same thing is happening on the right side for the BMI for that same boy. Basically, normally your BMI would slightly increase, but it certainly would not decrease when you're growing. So there's lots of reasons why we can think why that is the case. Maybe the kids are not able to absorb nutrients. Maybe they're not eating enough or not indicating that they need to eat, or they don't sense the hunger as they should. But we were quite interested to see whether we could see some of these deficits also in urine models, to get more at the systemic energetic basis of why we're losing weight with progression of the disorder. Next slide, please. And so in the lab at the time when we were doing studies, we had available these models that mimic MPS IIIA, the sulfamidase deficient mice, which have a mutation. They're hypermorphic alleles, and they mimic many of the, at least biochemical, but also some of the behavioral phenotypes that are associated with MPS disorders. And at the time, we asked a simple question. So let the mice age. The mice initially seemed to weigh more compared to the control wild-type mice, which are the mice that are unaffected. But that is because they have extended bladders. And the extended bladder problem in these mice is quite significant. The normal mouse has about 100 microliters of urine. But a sulfamidase deficient mice, on average, has 1 milliliter, which is 10 times more of urine in the bladder. So when we took the bladder out and we looked for the amount of adipose tissue, we were surprised to find that despite the organomegaly you would expect for the liver, that the amount of fat in the mouse was significantly decreased. And so here we call fat white adipose tissue, or WAT, and then it's either epididymal when it's basically around the reproductive organs, or visceral, and subcutaneous fat, which is underneath the skin of the mice. And then there is this particularly rare population of fat, which is brown fat, which is not so much a lipid storage organ, but more an organ that will use lipids to generate heat without having to shiver. But the long-term conclusion was that when we isolated the organs from the mice, that very clearly, and with progression of age with the disease it became more clear, is that they have a significant reduction of adipose tissue, to the point that we could hardly find adipose tissue in some of these mice. Interestingly, you could argue that maybe the loss of adipose tissue is a result of the lack of feeding behavior, but contradictory to what we see in the body composition of the mice ate significantly more than their wild-type counterparts. So they are eating more food, but somehow they're not able to store excess nutrients in the forms of lipid despite eating more. And energy balance is very, very simple equation. It's energy intake versus energy output. So somewhere in this tiptoe balance, there is a disequilibrium. And it seems like there is maybe a disequilibrium in the terms of how much calories we are burning in baseline conditions. So next slide, please. So this is kind of an important finding. And one of the things that's very striking is that both in obese patients, as well as in patients who can fail to make fat, which is what we're seeing in our MPS models, they both lead to metabolic dysfunction. They both are massive risk factors for insulin resistance and type 2 diabetes, but also for dyslipidemia. And with dyslipidemia, we mean the accumulation or the higher amounts of a lipid that is circulating in your blood. And the higher that is, the more likely you are to have cardiovascular events as well as type 2 diabetes. And so we're wondering, is especially the dyslipidemia phenotype also presenting in the mice because they don't have enough fat to store it away or to access it? So that's what we wanted to test next. And so we took these mice and we basically asked the question, do they have dyslipidemia? Do they have this altered lipid phenotype? So the next slide. And so when we take these mice, and they are preclinical models, and I do understand that, we could see that, in fact, with aging, the mice that were affected by Sanfilippo had elevated plasma lipid levels in the form of triglycerides. And it got worse with the age of the mice and much more clear. So lipid accumulation in the forms of triglycerides are often a result of what you're eating. So then we basically took the mice. The first graph here on the left is the fasting plasma lipid levels. So then we asked, is the response to a lipid load when you eat also different in the mice? And so we took these same fasted mice and we give them a fixed amount of corn oil. And then over time, we track the amount of lipids that accumulate in the circulation of the mice. And I hope you can see this here. So mice normally will never accumulate lipids in the liver, in the circulation, under normal conditions. So a wild-type mouse, you can see a very little increase in the right panel upon 60 minutes after feeding, but overall there is a very little change in the plasma triglycerides after a meal. However, much to our surprise, when we did the same experiment in the MPS IIIA mice, we saw a massive accumulation of lipids over time. And it was so striking that at the top, you can basically see that the plasma of the mice that you extract from the blood look milky. You could basically phenotype the mice by looking at their plasma. You had milky plasma because so much lipid was accumulating versus non-lipidated plasma of a normal control mouse. So that was fascinating. And so basically then we're trying to figure out whether this is obviously specific to the MPS IIIA mouse model, or whether this is like a hallmark of the MPS mice, that all of the diseases that mimic Sanfilippo to some degree have similar phenotypes. So the next slide. So this is where we worked with Patricia Dixon. She was at the time at UCLA, and she had in her colony MPS I and MPS IIIB mice, which are both different disorders of Sanfilippo disorder. And so we did exactly the same thing here. We gave the mice a fixed amount of corn oil, and you could see that depending on the degree of the severity of the mice's lysosomal storage disorder in this case, it correlated significantly with accumulation of lipids, with MPS IIIB mice having the highest amount of lipid accumulating that I've seen in a non-compromised mice before. So that gave us confidence that this dyslipidemia that we're seeing, and also actually the lack of fat accumulation in all these models, was something systemic or associated with lysosomal storage disorder, and something to pay attention to. So when there's something to pay attention to, that we try to figure out what the molecular mechanism is behind our phenotype. What is causing this increased accumulation of lipids in the circulation, basically? And so it's a very simple equation. So if you go to the next slide, the way I look at it is basically you have a container with a faucet at the bottom, and you have an inlet at the top. And either you have reduced distribution of lipids to the liver, adipose tissue, or the muscle, or you have increased input of lipids. More lipids are coming in, and because of that, you have a harder time to clear them more rapidly. And basically, to make a very long story short, what we figured out is that when we looked at the input of lipids coming into the circulation, there was something significantly different. And liver can produce lipids by itself under fasting condition. But again, when you're eating, what ends up happening is in the small intestine, and especially the proximal part in the duodenum, lipids are getting processed and absorbed and packaged into chylomicrons, which are lipid droplets decorated by proteins that help guide these lipids to tissues where they need to be either stored or used for oxidative purposes. So if you go to the next slide, we have these very elegant assays where we basically give the mice corn oil and at the same time inject them with a molecule that will prevent the degradation and uptake of lipids from the circulation. And what you get is a linear accumulation of lipids. And the linear accumulation is directly correlated with the capacity of the intestine to absorb lipids. And hopefully you can easily notice from this graph that the MPS affected mice absorbed twice as many lipids as the counterparts that were unaffected. The lipid levels are raised very high and very quickly over time, suggesting that there is some kind of energy deficit that is driving an increased needs for nutrients to be absorbed. However, because it's MPS disorders, we're always worried about the integrity of many of the organ systems, because the lysosomes are accumulating in almost every of the cell types in these disorders. So we're wondering maybe this increased lipid absorption is not a direct active process, but the fact that now the gut's more leaky because it's less tight, and so we actually asked that question. So to do that, so if you go to the next slide, we can do a similar assay. But now what you're feeding the mice is a very small molecule, which is dextran, and you can label that with the fluorophore. And normally very little of this fluorophore should enter the circulation in a certain amount of time. And so we feed that orally to the mice, and then we basically look at the amount of fluorescence that's accumulating over time, which is a direct measure. Oh, we're going too fast. We have to go back. Yes, thank you. A direct measurement of the amount of basically intestinal damage there is. If there's more intestinal damage, you'll see more fluorescence in the circulation, but we didn't. So it really meant that there was an active process going on, something instructing the intestine to absorb a lot more lipids because there is an increased need for energy. So then we go to the next slide. to figure out where are these lipids going could maybe give us an indication of where the energy deficit is. And so what we're doing here is we basically give the mice radioactive lipids, and after a fixed amount of time, we see where in the organ systems these lipids are accumulating. And very interestingly, what we're seeing is they're accumulating in the adipose tissues, and especially the adipose tissues that are thermogenic. So the brown adipose tissue is a thermogenic organ, and the white adipose tissue, especially underneath the skin, which we call SWAD, is also very thermogenic, suggesting that there was increased thermogenesis in the mice, which normally isn't needed. So if we go to the next slide, brown adipose tissue is not only present in mice, it's very abundant in infants, in the neck, but also in humans. And when you are exposed to a colder environment, you basically start building up more of that brown adipose tissue so that you can warm your body. And they're typically around your main size arteries, so they're keeping your blood warm. And here's a very interesting depiction of when you put a person in the summer or in the cold environment. You can now see more activity around the shoulder regions and the spine, and that correlates directly with the activity of the brown adipose tissue in humans, and as well as the energy expenditure going up. So next slide, please. There's different types of cells, and what makes these brown fat cells different is that they have more mitochondria. And instead of producing ATP in the mitochondria, they uncouple that process and they generate heat. They're basically heat-producing organs. And beige fat, which is in the subcutaneous fat, does something similar, but is more adaptive. And so here are some numbers where you can see that we have a bunch of brown fat as humans and that they burn a bunch of calories. But interestingly, when we go to the next slide, the amount of brown fat we have declines over time, and that correlates directly with the amount of visceral fat we accumulate because we create this energy deficit. And so the question was, is this downsizing of brown fat activity not happening in the MPS mice because of some mitochondrial dysfunction? So when we go to the next slide, please. And it's exactly what we noticed. The mice were always 1 degree Celsius warmer, which is very unusual for mice. That's the graph on the left. And on the right, we can measure heat production by the mice. And that was up as well, indicating that non-shivering thermogenesis was increased in the mice. So if we then go to the next slide, this was already shown by the previous speaker. This is our work where we basically then look at these different adipose tissues. And long story short is that you should see that there's less lipid accumulation in the MPS IIIA mice. It is much more red, it's much more dense in basically mitochondria, and the same is true for subcutaneous adipose tissue. So then you go to the next slide. So then we did microscopy, electron microscopy, and we looked at these mitochondria because, as already indicated, there is this indication that mitochondria in MPS mice and patients are dysfunctional. But what we really saw was a massive accumulation of very functional mitochondria, particularly in this brown fat. And that correlated directly with increased oxygen consumption, which is the bar graph I'm showing you below, which would make sense that a lot of these mitochondria are functional and driving increased energy need. So if we go to the next slide. So you can stain for mitochondria with autophagosomes, and what we think is basically the mitochondria are getting produced, but they're not getting degraded. And you get this overaccumulation of mitochondria in that thermogenic tissue that is driving increased energy need. And that's something you also see in some cancer cells, leading to cancer patients to become cachexic. And then on the right side here, you have some markers of mitochondria that are accumulating. They're higher, as well as autophagy. And so we're thinking that reduced mitophagy, which is the removal or the eating of mitochondria, is impaired. So if we go to the next slide. So we tested that by giving the mice everolimus, which basically promotes mitophagy. And so when we treat the mice with vehicle, which is untreated, we see again that there is increased mitochondrial accumulation in the MPS model. But if you go to the next slide, oh no, sorry, go back. Unfortunately, I forgot to remove the panel here. But if we treated, in fact, with everolimus, we make the MPS mice look exactly like the wild-type mice, and we kind of rescue that mitochondrial accumulation. So if you go to the next slide now, importantly, does this respond to peripheral enzyme replacement therapy? Can you reverse it? So we took these mice, we treated it with recombinant sulfamidase, and long story short is that the mitochondrial accumulation that you normally see in MPS was reversed. We prevented that overactive brown adipose tissue, and we were also able to reverse the dyslipidemia that's associated with that increased energy need. And so if you then go to my final slide, the long story short is that mitochondrial dysfunction or overpresentation, especially in mitochondrial active tissues, is something that is regulated by autophagy and impaired in MPS disorders, and can possibly also contribute to the wasting phenotype that you see in disorders. And the overall take-home message is that we need to pay attention not only to the obviously neurological aspect where mitochondria play a big role, but we should always keep in mind that treating peripheral effects of some of the lysosomal storage disorders go beyond the liver. They might actually affect energy metabolism in a significant way and can help the patients thrive also by considering not only treating centrally, but also peripherally. Thank you very much, and then we'll hopefully have some questions that we can answer with you on this topic, and here are all the people and the funding sources that were involved. I think somebody's on mute still.
Alexey, you're on mute.
I was eating apple and muted myself, restoring my energy balance. Thank you for both speakers for excellent presentations. And we have some questions. So I see there are two questions to Dr. Ganetsky, and one question is whether you think that the mitochondrial dysfunction or functional alterations in fibroblasts are representative, or they would be cell-specific, so for different tissues would be different.
Yeah, I think that's such a good question. One of the big challenges that we have is indeed that different tissues have different mitochondrial function. And the data that I showed you showed that across most tissues, in most patients, there's relatively high correlation. That, of course, comes from primary mitochondrial disease, where the mitochondrial dysfunction is a gene that's expressed in the mitochondria in most tissues. In other diseases, even in some primary mitochondrial diseases, the fibroblasts can have normal function, while other tissues that have more energetic demand than skin does have mitochondrial dysfunction. What tends to be true is that we almost never see the reverse. If you see dysfunction in fibroblasts, that's usually a very good marker that there's dysfunction in other tissues, because fibroblasts have so many backup metabolic and energetic pathways that if they're dysfunctional, that tells you that there's really a stress on the system that doesn't allow typical mitochondrial function in other cells.
Mm-hmm. Okay, and so then there was one specific question also, about your glutathione measures, whether you measure them in blood or in urine or both.
Yeah, so that's a good question. Glutathione is present in our blood, and there actually has, it really relates to the previous question of where do we look for things and how much does looking in one area, especially for someone who's interested in developing minimally invasive biomarkers, tell us about other areas of the body. We have glutathione in our blood, and what we've found, and really what the Stanford group has found, is that glutathione in red blood cells correlates with glutathione in plasma, correlates with glutathione in whole blood, and it does seem to correlate quite strongly with glutathione in brain, although that's a challenging question to ask because usually the tools that we have for looking at glutathione in brain aren't exactly the same as the tools that we have for looking at glutathione in blood, but they correlate. And so we look in blood, and we do think that that's a good marker of what glutathione has been in other tissues.
Okay, thank you. I had my own question for Philip. Actually, I was surprised by the difference between the MPS1 model and the MPS 3A and B models, because basically we expect that any lysosomal storage would block the autophagy. Do you think it's because the MPS1 mouse model is not representative and it's like less affected than human Hurler patients are? So
I think in that case you're right, and so the MPS 3B model was very affected and was very sick, and it's very clear, and it really correlates with the aggressiveness of the lysosomal dysfunction in the models you're looking at. So the worse it is, the more easier it is to pick this up. Yeah, absolutely. It's also an aging thing probably. So some mice, when you age them, they're more rapidly getting to that very pathological zone, where other ones take a little longer. Yeah, we noticed that. Yeah, if we age the mice longer, it would get worse as well. Yeah.
I wish the study could be expanded to other lysosomal disorders. And I also had this question for you regarding is anything known about dyslipidemia in the human Sanfilippo patients?
Yeah, so we tried. So with Raymond Chang at Chalk, we tried to basically dabble into this. The problem is that you're faced with control issues. So you're using the families often as controls. They're often children, and so what we noticed is that lipid levels range all over the place. And then some of the families had kids with metabolic syndrome that had very elevated plasma triglycerides, which were concerns for other reasons. So it's very difficult to trend that in a very young population, which most of the patients are. And it's also not even more difficult than then look at that in the older population. So it's not an easy study to do. We try to do it. There is some indication that it's sometimes higher, but sometimes lower. There's too much variability in the human population, which is the advantage of an inbred mouse model, obviously, where you can control these things a little bit better. So as a marker, I wouldn't say that's a really good readout for the dysfunction of mitochondria. But it's something to take into consideration when patients have this inability to gain fat accumulation, because lack of fat is definitely not good for metabolic diseases.
Right.
Anecdotally, I do hear of patients who have elevated lipids of one parameter or another, but yeah, it's hard to know quite what to make of that. And even fasting, lipid levels being abnormal. Yeah. And, as Dr. Gordts said, the cachexia, or, just this inability to maintain weight, and then the need for, additional caloric supplements or enteral feeding, is interesting because, when you're giving them so much calories already, prior to having swallowing problems, because I know we had that overlay of, the dysphagia, which then leads you to the enteral feeding. But even prior to that, there are challenges with weight gain that families face, and really dissecting that to understand the perhaps peripheral mechanisms of that aren't necessarily related to neurologic function, that maybe are modifiable, I think is really important, kind of information that you bring, that treating with enzyme or other drugs that actually could make you nutritionally sounder or healthier, or make your mitochondria, more supported, that perhaps there's benefit to that worth exploring in more research.
I fully agree. I think keeping peripheral mitophagy intact in these disorders, I think will be a huge benefit to the quality of life. The problem is obviously to do that centrally, which there you see more of these damaged mitochondria, as already been shown earlier. So it's going to be a fine balance, but it's interesting. It needs to be explored. I think you're absolutely right about that.
There was one question on this, actually, whether urolithin A can provide any health benefit, like treatment of whether you noticed anything in your mind.
Yeah, the only thing we tested was everolimus, which is a compound that will also promote mitophagy, and we did see there improvements in the brown adipose tissue, but we didn't really do the long-term study because of funding reasons, actually. But it would be interesting to look into that and see whether compounds that do promote that peripherally as well, that they can help with these peripheral issues that the patients are suffering from. But as far as I know, there's not. Again, as it was indicated as well by all the faculty here, is we need more research on mitochondrial dysfunction in this disorder, because I think it will be crucial to look at that in the future.
Thank you.
And I think there is a last question, and it's basically whether anybody think that that actually delay in the growth and in the bone growth, or specifically in the is maybe related to this energy deficit. Any thoughts on that?
That's an interesting question, but that would be hard, correlation to draw from that. There's no bone growth issues in the mouse models, just to put that out there. And maybe that's because, of course, the mice age really quickly and the bone growth is kind of already at a terminal phase when the real accumulation is happening. So this is a great question, and we will have to create different models to really address this, whether that's true or not. But this is a great question. Never thought about that, actually.
Well, we probably better wrap. This is one of my dream sessions, and I am so thankful to you guys for opening up this conversation to our community. Really important things to think about and a lot more work to do.
Yeah. All right. Thanks for having us. Thank you very much.
Thank you.
Yeah.
Thanks for having us.